Next up, we've got a great presentation from our science director, Dr. Eric Diehl. He's going to be talking about some of the TBRS community's research progress over the past year and some change, as well as our research roadmap. Eric has worked in epigenetic research for over 10 years. He completed his undergraduate and PhD in biology at the University of Western Ontario, and he then completed a postdoctoral fellowship in the laboratory of Rosanna Wexberg at Sick Kids Hospital in Toronto. And he focused on diagnosis of neurodevelopmental disorders like TBRS through methylation. He is also employed as a diagnostic laboratory geneticist at Sunnybrook Hospital in Toronto, and he lives in his hometown of London, Ontario, Canada. So please join me in welcoming Eric to the stage.
Perfect. Okay, thank you everyone for wrestling yourselves away from those posters. It was really difficult. I know there's a lot of really interesting stuff over there. We're going to have more time to look at them again later, so if your conversations got cut off, don't feel bad. You're going to get to finish them. So what I want to do in this little presentation I have is just sort of give you a sense of where we are as a community in terms of the research that we're doing and sort of where we've been, where we're going, and sort of what our goals are overall. So I'm a bit of a new face here at the organization, so I first wanted to tell you sort of what I'm here for and sort of what my job to do is and talk to you a little bit about it today. So what a science director does is basically maintain knowledge around enhancing the TBRS research network and research programs, engage with the patient community, meaning you all today, regarding current research opportunities to participate, build engagement strategies, and importantly, and what I'm going to talk about today, is conducting a comprehensive analysis to identify sort of missing components in our research program. So, one other thing I wanted to mention is just sort of, Kit introduced me and sort of my background here. So one of the things that I do is sort of the diagnostic laboratory reports that you guys get that sort of say, what the variant is and what the interpretation is. Like the other half of my time, my job is to write reports like that at hospitals. So if anybody has any specific questions about reports like that, I'm happy to answer them. We can talk about them. So what I want to talk about today is just sort of briefly give this slide that Jill presented at the last meeting, I think. And this is sort of the overall trajectory of the research roadmap in the TBRS community. So there's a lot of points here. I don't expect you to read all of them. Basically, we've started and done a lot of this work on the left. Let me get the pointer. We started by doing a lot of this work on the far left here, and we've transitioned to sort of doing a lot of this work here. We're sort of in this middle part right here right now of trying to identify biomarkers, trying to get new models up and running, trying to further define TBRS as a condition and sort of what the features are. And all of the talks today from all of the amazing scientists has sort of given you a good sort of snapshot of where we are, and I certainly can't do a better job than them in a couple minutes. But so what I want to talk about is sort of how we get to the next step, which is therapeutics and interventions and treatments, okay? So what are the possible treatments for genetic disorders? So when we talk about treatments and interventions, we think about a few different things. We think about drug repurposing, which is where you identify existing drugs. Sometimes that gets referred to as off-label use, where you find a drug that might be effective to treat some aspect of a condition. You take it from another disorder where it's approved and apply it to TBRS in this case. You can do high-throughput drug screening, which is when you have a whole bunch of drugs that you test one by one in like a model system, and you just look for hundreds and hundreds of them, and you say, okay, these 3 seem to be effective, and we're going to move forward and see if those maybe should go forward to a trial. There's rational drug design, which is where you specifically look at how DNMT3A works, and you say, we're going to design a drug that fits into this part of the bulldozer, to borrow Peggy's analogy from this morning, and sort of maybe get the cab working or get the, I don't know anything about trucks, the front part that lifts the dirt working. And then, so there's gene therapy. That's where you replace the whole copy of the gene, or maybe just a small snippet of the gene where the variant is, with a working copy. And then there's something that's a little more specific called ASOs, which you may have heard of, which are antisense oligonucleotide therapy, and that just is like something that comes in and tries to correct a really specific variant. So that's sort of what we're talking about when we talk about possible treatments. So one of the big parts of my job and why I got brought on was to do this sort of analysis called a gap analysis. And the idea of this is just to, it's a 4-stage process that looks relatively simple, sort of say, where are we? Where do we want to be? What are the holes on the way to get there? And then how do we implement a plan to bridge them? So in order to do that, I've been meeting one-on-one with all of the research in the network, many of whom are here today, some of whom are not able to be, and just having sort of conversations with them about their research and TBRS and where we're going and what we're doing. And the main thing I want to do today is sort of give you a summary of what everybody has to say and where people think we are. So there is really broad consensus that therapeutics are the goal and that we need more basic research to get there. So we know where we want to go, we know what the gap to get there is, and that is we need more basic science like is being presented today. Everybody also has enormously positive things to say about the biorepository and the registry, and that these are the best possible things the community can do, and you've sort of seen that today in all of the presentations have sort of been hammering that point home, and I just want to reiterate that. And something I was also sure to do in all of these meetings is sort of communicate and reiterate to all of the scientists the patient priorities, which Carrie did a great job presenting earlier, and so that everybody sort of understands what your priorities are, what your goals are, and that remains at the center. We really are big on patient-centered research here, and we want you all to be the ones who are guiding us in terms of what we want to do, okay? So that sort of remains at the forefront, and that's always being communicated. So to give you a really brief one-slide sort of snapshot, and then all of the individual researchers are going into more detail on this stuff, so forgive me if I missed anything. But I just want to say that coming in from an outside perspective, so I started here in July, the quality, the breadth, and the speed of the research in TBRS is really quite impressive. There's a lot of, so we were at the CZI meeting a few weeks ago in Los Angeles, where there's a lot of other communities like ours who are there with representatives talking about the challenges they're facing, and it really is amazing to see how much progress we have made in research in TBRS compared to other disorders, and really inspiring to see other disorders that are maybe 10 years ahead of us and the things they've been able to accomplish. So I just want to say, and to everybody on the team and to everybody who's here, like, it's really amazing and it's really exceptional what the community's doing. And I just think everybody needs to hear that this is really an exceptional community. And I'll just say that over and over again. So what have we accomplished? The patient recruitment to the research studies has been really great. We're doing that today, obviously, and that's making a big difference and is driving the research that you're seeing as we've been hearing about in our talks this morning. The mouse models that you've heard about a little bit, and you can sort of see the progress that those are making for us, and the outcomes that are coming from them have been a really big deal, and they're really important. And then other things that we might hear a little bit more about later are iPSC lines. Those are when you take, and something might be done on this with any of the cells here. I'm not sure exactly what our goals for them are, but you can take cells from one person and you can sort of backtrack them all the way back to being a stem cell. Then you can take those stem cells and turn them into any cell in the body. So these are really great for researchers who maybe want to create neurons in a petri dish, but obviously it's very hard to get neurons from a person. So you can use these iPSCs to create all different cells in the body, and those are a really great tool for research as well. So we're doing all of those things. We actually have an iPSC line we've developed that we're making available to researchers. So, okay, what do we think about the gaps that are here in TBRS research? So the ideal state, if I were to sum it up, would be to say that we want to have TBRS well characterized, with diagnosis being easier, and with there being clear management guidelines. And we heard all about that this morning, about what we need to do to get there. The other thing that I'll talk a little bit more about is that we want a therapy and an intervention, and which one is sort of how that works out of those 4 or 5 options I showed you. That's sort of still a question mark. So then the gaps to get there are, we need knowledge. We need to further define the TBRS phenotype, as Kate was talking about this morning. And for therapeutics, we need to understand what to target and when. So we'll talk about that first slide. This is largely just a reiteration of what Kate was saying this morning, so she did a much better job than I'll be able to, so I'll just go through it briefly. But at the diagnosis level, we have a lot of mutations or variants, those two words are interchangeable, that could be classified as pathogenic, and we've alluded to this a bit today, but they're not. So I know for many of you, you have a report that says you have a VUS in DNMT3A and are not sure, does that mean it's TBRS? Does that mean it's not? How sure are we? And that's really an unnecessary process for a lot of these variants. There's some problems in the way databases are curated for DNMT3A, and the clonal hematopoiesis issue that Peggy and Rachel were talking about this morning really complicates the way clinical labs classify variants, because you can imagine that figure that Rachel showed showing that there's older individuals that just happen to have DNMT3 variants. Well, one of the ways a clinical lab decides if it's that background variation that all of us have versus a variant that's pathogenic is to say, hey, do we see this in thousands of people that we've sequenced who are typically developing? And so we see DNMT3A variants in those people, and that makes clinical labs really hesitant to classify things as a VUS. And if we can put out some guidelines, if we can do some education for those clinical labs to give them the confidence that yes, this variant is causative of TBRS, then that'll go a long way to sort of reducing those number of VUS reports people get. So that's something I'm really interested in doing and something I think I can help out with. And I'll talk about that more in a sec. I'm actually going to skip ahead to that since I'm talking about it now. So there's a number of flyers over there that we've referred to. I want to call your attention to this one specifically with the National Brain Gene Registry. So Kit talked about this a bit earlier. What this is a registry where people with variants that are VUS or pathogenic in all kinds of different genes are being submitted to this registry, and they're trying to help figure out, using artificial intelligence and all kinds of really interesting methods, to sort out what variants are pathogenic and what variants aren't. And we're really far ahead of a lot of genes in that network. Many of them don't even have named syndromes. So they have a lot to gain from us, but we really have a lot to gain from them as well, because they're part of these networks that are involved in how these variants are classified. And you can really help reduce the number of VUSs that get reported in DNMT3A by submitting to this registry. And they're particularly interested in people with VUSs. So if you have a VUS, I would really strongly encourage you to do that. So I want to talk about the gap in terms of what to target and when. So I know therapeutics and intervention strategies are really important to a number of you. And basically what the message I want you to get is that we need to determine what treatment strategy is going to work, whether it's gene editing or all of those other ones I described. And we just, put simply, don't have enough data yet to know which one to focus on. We need to know, and I heard questions about this today, how early do we need to treat with a drug? How early is early enough? How late is too late? Should we be targeting DNMT3A, or is there something else we can target? Can we repurpose an anti-epileptic drug that might work particularly well in people with TBRS? These are just things we need a bit more knowledge in order to do. What I will say we don't have a gap in is an exceptionally engaged patient and caregiver community and really talented, focused researchers, and this meeting is really a testament to that. So we have all the pieces in place that we need. So this is sort of a picture to sort of sum that up. So we've got a bunch of different pathways to a bunch of different possible treatments. We have some gaps in knowledge in terms of, okay, how early do we need to treat if we're going to be doing a gene targeting event? So if we fill in these gaps, like researchers are doing here today, we can say, okay, this is the strategy that we want to focus on. We then have this arrow that's doing an enormous amount of work in terms of summarizing the work necessary to go from that towards getting to a clinical trial, but this is sort of where we are. So our priority right now is filling in these knowledge gaps in order to get us towards saying, hey, this is the treatment strategy that's going to work best for us. So the goals that I see that I want to help support and what we have as a community going forward, in the short term, we want to facilitate all of you in using the registries and repositories, for instance, Citizen, for instance, the BGR. We want to be getting funding to researchers, and we were talking about the money earlier today that Casey's raised to do that. And as we were saying earlier, we really want to get surveillance guidelines in place as soon as possible. And then in the long term, we want to support the identification, prioritization, and investment into therapies, as I've been saying, and explore some collaborations with other OGID communities to see if that can sort of support us to sort of get our numbers up. And all the time, we want to encourage collaboration and between everybody, between the researchers, but also between you and the researchers. It's really a two-way conversation that the patient community is leading. So with that, I just want to say thank you everyone so much for being here today, for being the leaders of this community. We really value and need your leadership. It's just amazing to see how focused and how driven everyone is, and it's really what's the basis for all of our progress. So just your continued engagement, as Terry Jo was saying this morning, your engagement with these surveys and all of this work we're doing really is producing demonstrable results that I think everyone should be really proud of. So thank you.